vocab. So Pacinian corpuscle carries on a spinal nerve. The sensory information represented on the skin by a spinal cord division is called a dermatome. Let me see if I can explain this to you so you can see where we're going with this. Everybody knows about the bicep. Is that fair? The bicep muscle that does flexion at the elbow. The nerve that supplies information to the bicep is a nerve called, you don't need to know this yet, I'm just trying to get you warmed up. This is a nerve called the musculocutaneous nerve of the brachial plexus. And it receives its information from the upper portion of the brachial plexus, C5 and C6 in the spinal cord. Now, that nerve that travels down to the bicep, you will know that damage to the spinal cord in the ventral horn, that's where the lower motor neurons are, right? You learned that already. You will learn that would be an upper brachial plexus injury if you had damage to the way the bicep is working. Now, if I ask you then, what dermatome is there on the anterior portion of the brachium? You would say a dermatome that is represented by C5 and C6. So the skin here, the Pacinian corpuscle here, that sensory information represented by C5 and C6 in this region of the arm delivers sensory information to my spinal cord at that level. The same level that brings the motor info carries the sensory info back. So when you learn the muscle and its innervation, you will have learned the skin dermatome. So in the nervous system, I'll show you a map of this. It's a useful map because somebody says, well, I've got pain in my leg, in my back leg, or along the lateral side of my leg. You think, well, they got a back injury. Where is that injury? Well, once you have learned the innervation of the bicep femoris, the long and the short head, you'll know where it's coming from, the spinal cord, and you can predict then. If you feel the pain here, that's sensory information, and it's on the same nerve that carried the motor. So I'll teach you the motor. By default, you'll be learning the sensory. Okay, so the Pacinian corpuscle is represented as a skin area, a dermatome. And that dermatome describes which divisions of the spinal cord are receiving that sensory information. Dermatomes, the area of skin, the dermatome is the area of skin represented in spinal cord divisions. So the Pacinian corpuscle from the dermatome, the skin area, carries information along the spinal nerve, and that information then travels through the dorsal root. There's the third big vocab. Through the dorsal root into the dorsal or posterior portion of the cord. Dorsal root, dorsal portion of cord. Remember, in anatomy, there aren't very many places like this, but this is one of them. There are no exceptions to this rule. That is always sensory information. Now, can you guys see on this picture? So I'm moving dorsal here. Can you guys see on this picture a neuron right there? You see a little lollipop looking thing on there? That is a particular kind of neuron called a unipolar neuron. And this is the only place in the body where you will find them. These unipolar neurons. So that's an important anatomy observation. Those are unipolar neurons. And they're clustered in this area. So that little lump that you see right there, that lump right there, is a cluster of neuronal cell bodies. How would it look in appearance to you? Gray or white? Remember, y'all can't, there's things you can never forget. They will never go away. They look gray or white. Jared?
Gray.
Why?
It's the bodies of the neuron.
It's the body of the neuron, right. If it's the axon, if the tissue is dominated by the axons, then they're myelinated. That means they have a white appearance. So this is a gray area here. And we call it, because it's a cluster of neuronal cell bodies, we call it a ganglion. It's really important. Write that down. A ganglion. So if there are 31 pairs of spinal nerves, how many pairs of ganglia do you think we have along the spinal cord? 31. 31 pairs of spinal ganglia. And each of these spinal ganglia is a cluster of unipolar cell bodies. Now, what happens, just so you can start putting all this stuff together, what happens if an individual bends over and picks up a heavy weight and they compress the fibrocartilage pad between L5 and S1 so that the annulus, can I use that word? So that the pulp protrudes out, poking the annulus out from between the bodies of the vertebral bones and pushing this dorsal root up against the transverse process. How will the body interpret that? That's sensory. And there will be pain fibers in there as well. Your body, what you will notice is this hurts because of the pain information in that dorsal root ganglia. The dorsal root ganglia, the spinal ganglia, that's where all the sensory information is. The spinal ganglia or the dorsal root ganglia, a cluster of neuronal cell bodies, 31 pairs of them. That information then comes into the spinal cord on the dorsal side and then turns and goes toward the brain. Now I'm going to fill in the details of this as we go through the semester. But for today, learn the general sequence. From the spinal cord, the information passes to the brain stem, and that would be through the medulla oblongata, the pons, and the midbrain, ultimately delivering that information to the thalamus on its way to the cerebral cortex. So here's the pathway, the general pathway. The Pacinian corpuscle sends information along a spinal nerve through the dorsal root ganglia into the dorsal portion of the cord. Pacinian corpuscle, spinal nerve, dorsal root ganglia, dorsal portion of spinal cord. Then that information passes through the brainstem, medulla, pons, midbrain, on its way to the brain. Medulla, pons, midbrain, through the brainstem. This is brainstem right here, medulla, pons, midbrain. That information arrives then, its second order neuron arrives at the thalamus. The thalamus is a major relay station. We'll say it over and over and over and over and over. The thalamus. Everything that comes into us goes there. It's like Grand Central Station for sensory information. The thalamus then delivers information into the cerebral cortex, where conscious perception is. So there's a base level delivery of sensory information into the nervous system. We call this sensory information, we call this afferent, A-F-F-E-R-E-N-T, sensory or afferent information traveling to the central nervous system. Use the skin receptors to do it. Now, this is not the time for the details of this, but students have in years past said, I don't understand. I understand what you mean. It's traveling to my brainstem? It's going up the spinal cord to my brainstem? Yes. And later in the semester, this is a transverse section of cord. I'm going to show you where that information is. Different kinds of sensory information are found in different places in the spinal cord. So a good anatomy student can look at an image of a spinal cord that has a lesion on it or damage to it. And you can predict, knowing this, what the sensory deficit is or what the motor deficit is, or both. by knowing where this information is located. And so the pathway that I just showed you is a pathway called the lateral spinothalamic tract. And so it has, these bundle of axons are located in a place in the spinal cord where we can locate them. So this is something to look forward to. Maybe in your life right now you're not looking forward to it. But this is the kind of anatomy stuff that explains things. I look at a spinal cord injury, you tell me it's an injury at L2 to L4, and you show me where that injury is in the lateral funiculus, I can tell you exactly what this person is experiencing. Can they move their muscle? Which muscle can they move and not move? Are they feeling pain? Where is that pain they're feeling? So this is coming in your lives, the details of where this information is in the spinal cord. All right. Now, this one shows up 2 or 3 times. In this presentation, it shows up multiple times in the nervous system presentation. It shows up multiple times in physiology. This is the first time during the semester you've ever seen it from me. It comes later in your presentation because it's motor. I'll grant you it's motor. But I just used the ganglia. I thought this morning, wait, when you mention ganglia, just show it to them one time. Get it in their heads. So when you do the details of it later, they'll be working on it already with you. Ganglia. I said ganglia, right? Didn't I say that here? Dorsal root ganglia, okay? Now, when I describe the motor information going out to some structure, I will use this ganglia language with everything I say about the autonomic nervous system. So if I'm going to go out to a sweat gland, an eccrine sweat gland, or I'm going to go to the heart, or I'm going to influence smooth muscle in the blood vessel walls, or smooth muscle in the digestive system, I'm going to do that with the autonomic nervous system. And when I do that, I'm going to talk about the ganglia, or the ganglion. And now you know what I mean when I say that. The ganglion is a cluster of cell bodies. And as it turns out, there are dorsal root ganglia on the spinal cord that carry all of the somatic sensory stuff to my body. And I just taught you that, the general pathway. But coming in your lives, I'm going to teach you the motor output for going to these autonomic structures. And I'm going to use ganglia language with that. As we go through anatomy, I'm going to locate where these ganglia are. So there are lots and lots of them. And I'm going to point them out to you. Where are they located? What does it mean to have various issues in various parts of the body involved with heart and digestion and glandular secretion? It is the language of autonomic ganglia. So I want to show it to you here. If we define sympathetic or parasympathetic, we're going to define those, the basic anatomy of them, based on their ganglia, the clusters of cell bodies where the information is being processed. Okay. That's an aside. All right, here's a look at the receptors. We'll finish here today with these 2 slides on the receptors. In the skin, we say that there are both unencapsulated and encapsulated sensory pieces. There are, for example, as I mentioned to you, free nerve endings within the skin. Free nerve endings unencapsulated. They're just raw nerve endings that are found in the epidermis. And so this picture tells you that these guys are also found in ligaments and tendons. They're also found in the dermis. Free nerve endings in the dermis, is that right? You've seen that already? Yeah. In the epithelia, in the epidermis, in the cornea, on the outside of your eyes, mucosa and glands, they're free nerve endings. There are also modified free nerve endings. Some debate their importance, and some will even call these encapsulated. These modified free nerve endings we called Merkel cells. Do you recognize them? The Merkel discs. I pointed these out as one of the cell types in the stratum basale of the skin, in the basal layer of the epidermis. And hair follicle receptors in and surrounding the hair follicles. Have you seen that? I pointed that out to you a couple of times. Now, there are also encapsulated receptors that you've seen already, and I shouldn't have to say it now. Meissner's corpuscles, where are they? They tend to be closer to the dermal papillae, right? And where are the Pacinians? They tend to be deeper in the dermis, okay? So there's a little bit more in terms of detail about where they are that you should know. Meissner's corpuscles are not as abundant as the Pacinian. you can see here that they are located normally in the dermal papillae of hairless skin. So there's not a lot of that on our bodies, but these would be particularly in the nipples and the external genitalia, on your fingertips, on the soles of your feet, on your eyelids. You find these touch receptors. And they are light pressure receptors, these Meissner corpuscles. What I have really had to say that, if you know that the Meissner corpuscle is in the dermal papillae, and I asked you, which of these two is more interested in light pressure versus deep pressure, Pacinian and Meissner? If you know where they are, you don't have to memorize that. The Meissner corpuscle is closer to the edge, so it's light touch. The Pacinian corpuscle is deep in the dermis. It's for deep pressure, stretch, vibration stuff. So these guys are found, as we've said, in the subcutaneous tissue of the skin. They're also in tendons and ligaments, and they're also in the fingers and the soles of the feet, external genitalia. These are more abundant, these Pacinian corpuscles. That's why it was on that nervous system picture as a prototype. Meissner's, and there's one more. Meissner's Pacinian, and there's one more. These guys, these Ruffini corpuscles, or Ruffini end organs that detect continuous touch or pressure. So, very light touch, Meissner. Very deep touch, Pacinian. Ruffini, how do you continue to get information? These guys will adapt. The Meissner's and the corpuscle, the Meissner's and the Pacinian, you know, my watch is on my arm, my glasses are on my head, I forget. They adapt. Rapidly adapting. Rapidly adapting. The amount of information going, once you've hit them, will stop after a while. You can't continue to hit them. So the way you continue to get information is with these guys, these Ruffini's corpuscles. They're found deep in the dermis and the hypodermis and in the joint capsules. And look, these guys are very slowly adapting receptors, the Ruffini's corpuscles. So I always think about, I forgot that my hat was on my head, my glasses were on my face, my watch was on my wrist, because Meissner's and Pacinian adapt. They'll quit sending action potentials after a while. The Ruffini's corpuscles don't. They will continue to give you information about what is being touched on your body. So they play different roles. And I would like for you to know where they are. So Ruffini's corpuscles tend to be deeper in the dermis. The shallow ones are the Meissner's. Okay, so I used then I used the skin receptors as an excuse to teach you a little sensory pathway in the nervous system. So that was your first introduction into nervous system. I'm going to tidy up on Wednesday, fingernails and hair anatomy, and then I'm going to come back to the nervous system again. So my job, here's my job for the next, 3 or 4 class periods. I've got to teach you the basic anatomy, the rest of it, of the nervous system. So I've done my sensory piece now. So I've got the motor stuff yet to go with you. So we'll review the sensory as well. And then I've got to teach you the basic anatomy of the vascularization. You got a little bit of this already. You know about the simple squamous epithelium. You know about the smooth muscle in the blood vessel wall. So we just got to tidy that up a little bit. Differences in the anatomy of arteries and veins. Because you're about to start touching them. And then, the last thing I got to do is I got to teach you the basic anatomy of a muscle. I want a student of mine, before you get really into the muscle dissections, I want a student of mine to be able to describe everything I am touching. From the fascia and their names into the individual muscle cells, and break it all the way down to the proteins. So that'll be the last thing that we do. We'll break the muscle all the way down. From the epimysium all the way to the interaction between the myosin and actin inside the muscle cell. Then we can move into the lab. We can go after these structures. And I can start at the head now and begin to teach you the muscle attachments and their innervations, and we'll move through the body that way. That's the logic of this class. All right? until Wednesday. Tidy up skin on Wednesday. Fingernails and hair.